Hello, everyone. For my SciComp project, I'm going to be teaching you all today about how energy producing processes in our bodies can regulate brain development. So every day you eat to survive, converting this fuel into the energy that powers the complex processes of the 30 trillion specialized cells that form all the tissues of your body. It is important for all the programs that convert food into energy to function properly throughout your life, especially while you're still developing. Issues with the function of these programs are expected to cause various developmental diseases, particularly brain disorders like autism and ADHD. So early in brain development, the brain consists of mostly of a cell type called radioglia. You can think of these cells kind of like college students. They're exploring a bunch of different paths right now, but they haven't committed to a final career or fate yet. So radioglia can become cells like neurons that transmit and receive information, or astrocytes that play a supportive regulatory role in your brain to help it function as it should. But interestingly, these adult cell types preferentially utilize different energy producing pathways, as do the radioglia that they come from. So in developmental time, neurons come before the astrocytes, so I'm just going to focus on those. So let's just say that radioglia majorly use pathway 1, and neurons use pathway 2. So let's say pathway 1 needs burgers to function, while pathway 2 needs chicken tenders to function. So if we are at a point in development where radioglia should be majorly proliferating and not choosing their fates yet, but we barely have any burgers and we have a lot of chicken tenders, the lack of burgers may lead radioglia to become neurons sooner than they intended. The availability of different fuels and environmental conditions needed for these pathways is expected to affect how exploratory radioglia choose their final form and the timeline of this transition. This is a possible explanation for brain development disorders because any changes in the pathways used may impact when and what these radioglia cells become, thus impacting the structure and function of the brain. But in order to understand this question, we need to study the development of the human brain when differences in energy program usage are present. Since it's clearly not ethical to study and experiment on a developing baby, we can use some alternative methods. So one of these emerging methods is called organoids. So organoids are made from cells that are pretty similar to radioglia, except these ones can become almost any cell in the body. Meanwhile, radioglia can mostly become the neural lineage cells. So we start out with these cells that can become almost any cell, and we grow them, and then we aggregate them in a V-shaped plate, and this allows them to take on a 3D shape and be a 3D model for us, which is really unique. So then by supplementing these non-specific 3D models with specific molecules and nutrients, we can prompt them to become radioglia, which then become the other cells of the brain. So not only does this model allow us to view brain development over time, but it gives us an idea of the organization of the cells and their interactions in the brain. So organoids are typically cultured in high glucose, high oxygen environments, and this doesn't really accurately match the environment of the developing brain. It's really the opposite. Um, so since glucose and oxygen are important nutrients that are used differently between energy producing pathways, just like the burger and chicken tender example, we can look at varying conditions to see how they impact the development of our organoids. So we have our typical condition, and then a condition with low glucose, low oxygen that more closely represents the developing brain. And then we have some in-between conditions as well. So this will allow us to see how essential are appropriate nutrient levels to brain development, and how is the utilization of energy producing pathways altered based on this. And then how specifically is the development of the brain altered in the terms of the kinds of progenitor cells or the kinds of these radioglia, and then when and where they transition into neurons and astrocytes and all the other cells of the brain. Thank you for listening.